Hello everyone, I'm going to begin the Lab 3 overview for 1408. Lab 3 is pretty straightforward as long as you read the information and review the information it asks you to review, but it's acids, bases, and pH lab. Talked about in class in chapter 2 how when you talk about acids, bases, and pH you have to keep in mind you're talking about in a solution, okay? And we define a solution as a solution being formed by using a solvent. In this case, the solvent is always, in this class, water, okay, plus dissolved solids. What water is capable of breaking down, okay? So here, showing a saline solution or salt solution being created by adding a teaspoon of salt. Polar water molecules are able to break the ionic bonds, creating this saline or salt solution, okay? Now, when we talk about acids and bases and pH, we also talk about compounds that sometimes are referred to as ionic compounds. Okay, these compounds are a key chemical bond that forms them. It's an ionic bond. And when you add a compound that's formed through an ionic bond, when you add it to water, it undergoes a process called ionization, resulting in the release of a cation and an anion. So that's something we'll have to pay attention to, that you have to pay attention to as I go through this overview. Now, when you talk about pH, you talk about measuring the pH of common household solutions, which this scale is showing you, and I gave you a little different diagram in your lab handout in the protocol, but it's still a similar concept, and it says the pH itself, the best way to describe it, and I said this in lecture, is pH you can think of as the potential for protons in a solution. Now, we call these H pluses hydrogen ions or protons. Okay, and it shows you here on the pH scale, but it gives you a visual over here. If we ever talk about a solution having a neutral pH, one way to describe it is that the number of protons and OH negatives, called hydroxide ions, are equal. Okay, but if you start adding a chemical that causes the number of protons to increase in a solution, but causes the number of OH negatives or hydroxide ions to decrease, okay, then we say we start to create an acidic solution once the pH drops below 7. So the pH has to drop below 7. However, if we add something that causes the removal of these protons, causing a decrease in the concentration of protons and inversely an increase in the number of OH negatives or hydroxide ions, once the pH goes above 7, we say we have a basic solution. So bottom line is, okay, the pH and the concentration of protons are inversely related. Okay, so we use this bracket to represent concentration. This H plus, remember, it's called a hydrogen ion, but it's also called a proton because it lost the only electron it had. So if the number of protons are increasing in the solution, then we say that it's decreasing. Sorry for the bad color scheme here. Likewise, if we say the number of protons in a solution is decreasing, I'm going to have to put it down here because I didn't leave room, is decreasing, then we say the pH is increasing. Now don't directly go to your right and compare this because I've actually got these reversed. I should have written this on the bottom up top. Okay, so over here, once the pH goes above 7, from neutral, this is what's happening. And crisscrossing that over here, once the pH drops low enough before 7, we say we have an acidic solution. But initially, simply the way it works, concentration of protons are increasing. Okay, concentration of protons are increasing as the pH decreases and vice versa. Then we talk about inorganic compounds called acids and bases. The key chemical bond forming an inorganic compound called an acid or base is an ionic bond, an ionic bond. So the only acid you need to know, specific acid, just like in lecture, same for lab, is acetic acid. Acetic acid, if you look down here, okay, it's formed through a covalent bond. It's formed through an ionic bond. God almighty, I keep messing up, sorry. It's formed through an ionic bond that holds this proton to this chlorine anion. And when you add this acid, HCl, okay, to a water-based solution, it undergoes ionization. It results in the release of protons and chlorine ion. Well, any inorganic compound that releases protons into a solution, causing the pH of the solution to decrease, it is considered an acid. Okay, so acids, okay, can create acidic solutions by increasing the concentration of protons, thus decreasing the pH. Solution itself is that the acid is being added to. This is a solution that the acid is being added to. This solution, though, won't be considered acidic till the pH falls below 7. So sometimes we describe an acid such as HCl as a proton donator or hydrogen ion donator. For the base, it's just the opposite. Here's a base, the only base you're responsible for knowing, just like in lecture, sodium hydroxide. Okay. Sodium hydroxide is actually two chemical bonds that contribute to its formation, but the key chemical bond again is an ionic bond, holding the sodium cation to this hydroxide anion, negatively charged, OH negative. Now, a base is any compound, okay, that when added to a solution can remove protons from that solution. Okay, so a base will remove protons from a solution, causing a decrease in the protons, thus an increase in pH. However, the solution itself is not considered a basic solution until the pH falls below 7. Okay, what will happen is this hydroxide ion, once it's released in solution, like up here, okay, it'll go over to wherever it sees any protons, and it'll chemically react with them, simply forming a molecule of H2O. Thus, you can say remove, some people say neutralize if you want to be technical. But in either case, it's causing a decrease in the number of protons, which initially causes an increase in pH, and if the solution the base is being added to finally causes the pH to go above 7, I think I said below before, but above 7, then it's a basic solution. Sorry for all this, I'm kind of rushing through this. Then there are chemicals that are not acids and they're not bases. They're called buffers. Okay, the purpose of a buffer. Well, first of all, a buffer is solution, and it functions in minimizing changes in pH, okay, when a base or an acid is added to that solution. Companies make different types of buffers, okay, that can be used in labs for research. There are buffers they make that when you add an acid or a base to that buffer, it'll keep the pH at a 7. There are other buffers we use in labs where you add an acid or a base, well, it's supposed to keep pH at 4, an acidic pH, and it'll keep it there. There are other buffers that we use that are supposed to keep the pH at a basic pH, just 10, no matter what you add, acid or base. Well, a buffer is not an acid or a base. It's actually a mix of a combination, okay? So the buffer is actually a solution that's a mix of a combination of a weak acid and weak base, okay? It has both of these chemicals, okay, to help offset if you happen to add an inorganic acid or base. So this example here is an example of a buffer that's supposed to maintain pH 7. If you were to add an acid such as HCl, hydrochloric acid, to a buffered solution, a solution that has a buffer and it's supposed to keep the pH at 7, every time this acid releases proton, protons will chemically react with the weak base, and this is what will happen. So what we don't get is an increase in the concentration of protons, and since HCl cannot cause an increase in concentration of protons because this part of the buffer removes the protons, the pH stays at 7. Now, if you were to keep adding HCl and keep adding it and keep adding it, eventually the buffer would run out of weak base. In other words, eventually you would saturate the buffer or overwhelm the buffer, and then yes, the pH would begin to drop, but it would take a lot of acid. In person, we would have done that, but we don't do it here for our virtual lab. Now, why do we talk about acid bases and pH in biology? Well, because there's physiological relevance to it. It has to do with our body. Okay. Our blood pH normally ranges from 7.3 to 7.45. In lecture, I give a pH of 7.4, which is roughly an average. But this is actually the range of our blood pH. This is considered normal blood pH. All right. Well, keep in mind, you're constantly breathing. You're inhaling O2 and you're exhaling CO2. The O2 that you're inhaling, okay, we'll talk later in chapter 6, is being used by cells of your body to produce energy. But when your cells produce that energy utilizing O2 and nutrients, they produce a waste product. That waste product is carbon dioxide. Okay? And what happens is in order to get rid of that CO2, your cells, which produce this waste product CO2 while they're making the energy they need, that CO2 will move out of the cells into your blood. Okay? roughly 55% of your blood is made up of plasma, okay, which is a water-based solution. And what will happen is the CO2, okay, will react with the H2O in your blood, creating an acid known as carbonic acid. Okay? But our body has a carbonic acid buffering system. Remember what I said a buffer is? The job of a buffer is to maintain a set pH. So our body has a buffering system. This buffering system is built into your blood to ensure that when this carbonic acid is formed, okay, if it functions like an acid, releasing a proton, okay, into the solution part of your blood, okay, what will happen is this buffering system will help ensure it has what it needs to push the reaction back here. So the formula that is given for what happens to the CO2 in your blood is this here. Carbon dioxide will, I'm sorry, let me just, God, that was bad. CO2 will combine, okay, with the water in your blood, forming carbonic acid and then releasing a proton and leaving a molecule that is considered an anion. I'm not going to test you on it, but it does have a name. It's called a bicarbonate anion. But what will happen is this reaction, with the help of this buffering system, will be pushed back to the left. Okay, so these arrows that you see here, and here that are pointing both ways, are to indicate, okay, when this carbonic acid system is buffering system is working properly, these arrows signify reversible arrows in chemistry, meaning that the CO2 will react with water going this way, releasing a proton and a bicarbonate anion, but the reaction will reverse and go back this way. Therefore, we never truly get an increase in the number of protons in our blood. It's buffered. Therefore, the carbonic acid buffering system helps keep our blood pH here within a normal range. Okay. In the lab you've been given, it's going to give you a scenario. It's going to say, hey, look, in this scenario, let me use a different slide here. In this scenario, I'm going to use the last slide. In this scenario, they're going to say, hey, look, we're going to give you a beaker. I know this is an ugly beaker. We're going to give you a beaker of water. And then what you're going to do through a straw is you're going to introduce carbon dioxide into that water. Okay? It's going to say you have a straw and you're going to exhale into the water. Well, what gas are you exhaling? Why are you exhaling? To get rid of CO2. Except in this setting, okay, it's not like your blood. There is no buffering system. So in this case, when you introduce CO2 to the water, the carbon dioxide is going to interact or react with the molecules of water. Okay? There's no buffering system present. All right, so when the CO2 reacts with the water, unlike your body, the reaction is going to go one way, to the right. I'm going to let you fill in the rest. I'm not going to give you the rest because it's up to you to answer the questions, which are on the last page. So I'm going to pull this document up. Hold on, I'm trying to pull it up. Sorry, this isn't going smooth. It was meant to go smoother. So in your lab handout, it gives you some objectives. Make sure you read those, and it gives you a video, and it tells you to look at your lecture notes. So this lab is a good practice run for you to study for Thursday's test when it comes to pH. Gives you the table here, talks about different ways to measure in pH, and then this here, it'll be on page 4. Okay, this is the point where you're going to have to go over this thoroughly. Everybody, don't just try to jump in and answer the questions. Read through the lab, go through everything so you know how to answer the questions. Read through the experiment thoroughly. And then what you're going to submit is on the last page. Okay, you can do it on this paper and insert it. Just make sure you make room. Don't squeeze it in handwriting it. I got to be able to read it or I'm not going to grade it. Okay, or you can type and retype the questions and place the answers on a separate doc and then upload it. But your lab report in the end will consist of answering these questions. But in order to answer them correctly, you need to make sure that you read the rest of the lab first, okay? All right, that's all for now. I'm going to stop the recording, and y'all take care.